I am Professor Rajan from India. This keynote presentation is on virus genome analytics. This presentation would not have been made possible without substantial technical support from Dr. Andres Valionis, Dr. Jean-Claude Perret, Dr. Michael Patrick Coyle, Dr. Manish Prateek, Dr. K. Janardhanan, Mr. Satya Govindarajan, Mr. Prashanthi Govindarajan, B.N. Badri, and Dr. S. Geeta. Some of the pertinent research has already been published in global journals, New York, and London Journals Press, United Kingdom. This presentation will be made in three parts. Part 1, virus genome analysis in terms of percentage nucleotide concentrations. Part 2, virus genome analysis in terms of JCP golden ratios. Part 3, a feasibility study on dismantling virus structures using sonic frequencies. Let's begin with part 1. Now I shall define percentage nucleotide concentration. Analogous to the notion of pH value of a solution, the definition of percentage nucleotide concentration of a genome sequence is considered here. Given a genome sequence, the number of a particular nucleotide, say adenine A, present in that genome sequence is counted and the sum is divided by the total number of nucleotides in that genome sequence. The fraction when multiplied by 100 yields the percentage concentration of adenine PA. Similarly, one can evaluate PT, PG and PC. The values of PA, PT, PG and PC of a genome sequence and a possible combination of these values, like the proportion PA is to PT is to PG is to PC, seems to pave a way to classify and characterize genome sets. 9 virus genome data shown here were obtained from NCBI website and they are used for analysis. Percentage concentrations of adenine, thymine, guanine and cytosine were calculated and results presented in the table given in this slide. From this table, we observed that PA values of genomes 1, 2, 3 and 4 almost remain the same. Similarly, PT values of these genomes almost remain the same. PG values almost remain the same and PC values also remain the same. Genomes 5, 6 and 7 exhibit a similar property. Genomes 8 and 9 also exhibit similar property. Now, let's study the pairwise comparative analysis of percentage concentrations. First, we see for each genome what is the change in percentage nucleotide concentration has occurred. The change in the percentage nucleotide concentration can give a statistical overview of how the percentage distribution of nucleotides changes from one genome to another. A positive value in the following graphs, which I am going to show now, depicts an increase in the concentration from genome X to genome Y. A negative value depicts a decrease in the concentration from genome X to genome Y. Differences in percentage nucleotide concentrations with respect to genome 1 and remaining 8 genomes are shown in this graph. In addition, absolute differences are also shown in the figure. Differences in percentage nucleotide concentrations with respect to genome 2 and remaining 8 genomes are shown in this graph. In this slide also, differences in percentage nucleotide concentrations with respect to genome 3 and remaining 8 genomes are shown in this graph. Differences in percentage nucleotide concentrations with respect to genome 4 and remaining 8 genomes are shown in this graph. Please look at this slide. Surprisingly, we see a reverse behavior in the differences in percentage nucleotide concentrations with respect to genome 5 and remaining 8 genomes which are shown in this graph. Here we see the reverse behavior of the differences in percentage nucleotide concentrations with respect to genome 6 and remaining 8 genomes which are shown in this graph. Once again, we see the reverse behavior of the differences in percentage nucleotide concentrations with respect to genome 7 and remaining 8 genomes which are shown in this graph. In this case, we see reverse behavior of the differences with considerable differential errors in percentage nucleotide concentrations with respect to genome 8 and remaining 8 sequences which are shown in this graph. Here also we see reverse behavior of the differences with the considerable differential errors in percentage nucleotide concentrations with respect to genome 9 and remaining 8 sequences which are shown in this graph. Some important observations what we have made. Like the genome sequences 5, 6, 7 and 8, genome sequence 9 also shows almost a reverse characteristics when compared to other genome sequences 1, 2, 3 and 4. Thus, genomes 1, 2, 3, 4 are of one type and genomes 5, 6, 7, 8 and 9 are of another type. One may observe that the differential errors are found to be minimum in the case of genome 5, genome 6 and genome 7. Alternatively, the differential errors are found to be considerably noticeable in the cases of genomes 8 and 9. Now we move on to part 2, where virus genome analysis is carried out in terms of JCP golden ratios. 130 complete genomes of various categories of viruses are drawn from NCBI website for data analysis. All 130 virus genomes have been listed in these 2 slides. Now let me briefly explain the notion of golden ratio in case of geometric figures. The term golden ratio, GR, refers to a number mostly observed while taking ratios of distances in simple geometric figures such as pentagon, pentagram, decagon and dodecahedron. It is usually denoted by the symbol phi. Figure A shows a rectangle having sides in the ratio 1:x. Now, for a unique value of x, which is denoted as phi, partitioning the given rectangle as a square and a small rectangle as shown in figure B, the smallest rectangle will have its sides in the same ratio 1:x. One can further partition the small rectangle into a square and a smaller rectangle. This kind of partitioning could be carried out indefinitely. One can observe smaller rectangles have their sides in the ratio 1:x in every partitioning. The rectangle shown in figure A is called a golden rectangle. This property of rectangles having sides in the ratio 1:x is called golden ratio property. The unique number is x is equal to 5 is equal to 1.618. Jean-Claude Perret proposed a method of attributing a quantification measure on the lines of golden ratio. Golden ratio GR of a viral sequence may be treated as the solution of a rational polynomial x1a plus x2t divided by x3g plus x4c, where x1, x2, x3 and x4 denote certain coefficients. A denotes the number of adenines in the sequence. T denotes the number of thymines in the sequence. G denotes the number of guanines in the sequence. And C denotes the number of cytosines in the sequence. In order to honor Jean-Claude Perret, we denote this golden ratio as JCP golden ratio. Golden ratio of a viral sequence obeying natural pairing could be treated as a solution in the rational polynomial x1a plus x2t divided by x3g plus x4c, where x1, x2, x3 and x4 denote certain coefficients. We assign values like x1 is equal to 1, x2 is equal to 2, x3 is equal to 3 and x4 is equal to 4. We call this assignment as A1. 3 more assignments are made as A2, A3 and A4, which are cyclically permuted versions of A1. Similarly, one can have A5, the graphically inverted version of A1, and its cyclically permuted versions A6, A7 and A8. JCP golden ratios are calculated for all assignments and results shown in the graph. In this slide, we see golden ratio values for the first 10 genomes. We observe that assignments A1 and A7 yield almost same GR values. Assignments A2 and A8 yield almost same GR values. Assignments A3 and A5 yield almost same GR values. And assignments A4 and A6 yield almost the same GR values. This procedure is repeated for all 130 virus genomes and GR values calculated. In all the cases, the assignment pairs A1, A7, A2, A8, A3, A5 and A4, A6 independently show almost equal GR values. Pairwise comparison has been made for various assignment pairs and results indicated in graphs. GR values of all 130 virus genomes remain almost the same for A1 and A7. GR values of 130 virus genomes remain almost same for A2 and A8. GR values of all 130 virus genomes remain almost the same for A3 and A5. And GR values of 130 virus genomes remain almost the same for A4 and A6. The same exercise is repeated for unnatural pairing of nucleotides also. Golden ratio of a viral sequence obeying unnatural pairing is a solution to the rational polynomial x1a plus x4c divided by x2t plus x3g. As usual, x1, x2, x3 and x4 are coefficients. Whatever features exhibited by 113 genomes while calculating GR values using natural pairing of nucleotides are also exhibited by the 130 genomes while calculating GR values using unnatural pairing of nucleotides. As it is done in the case of natural pairing, pairwise comparison has been made for various assignment pairs and results indicated in the graph. GR values of all 130 viral sequences remain same for A1 and A7. GR values of all 130 viruses remain same for A2 and A8. GR values of all 130 viruses remain same for A3 and A5. And GR values of all 130 virus genomes remain almost the same for A4 and A6. The question that arises here is whether one can use different numerical values for assignments. One cannot have random values assigned in the coefficients. Let us recollect the value of 5 in the case of golden rectangle. However, it has been found that one can use selectively prime numbers for all 8 assignments. This exercise of evaluating JCP golden ratios for all 130 virus genome sequences for A1 is equal to 1357 and its cyclically permuted versions A2, A3, A4, the graphically inverted version A5 and its cyclically permuted versions A6, A7 and A8 has been repeated and results compared with the previous ones. As per the definition, JCP golden ratios, their values are expected to remain within certain limits, irrespective of the numerical values assigned to the coefficients in the formula x1a plus x2t divided by x3g plus x4c. Now, the error values obtained using the formula a of 1234 minus a of 1357 yield interesting results. The error values of a of 1234 minus a of 1357 are calculated and the values for the first 33 virus genomes are given in this slide. From graphs, one may observe that the error values obtained using the formula a of 1234 minus a of 1357 yield very minimum differential errors. In this slide also, one would be able to see very minimum differential errors. This means that various numerical assignments do not cause any deviation in the golden ratio property. From figures, one would observe that virus sequences S26 and S32 show considerable errors in their GR values. That is A1 of 1234 minus A1 of 1357, A3 of 1234 minus A3 of 1357, then A5 1234 minus A5 of 1357 and A7 of 1234 minus A7 of 1357. These error values are given below, highlighted in red color. Alternatively, A2 of 1234 minus A2 of 1357, A4 of 1234 minus A4 of 1357, A6 of 1234 minus A6 of 1357 and A8 of 1234 minus A8 of 1357 do not show much errors in their GR values for S26 and S32. Once again, with reference to figures, one would observe that many of the virus sequences in the band S88 and S130 amounting to 43 genomes show considerable errors in their GR values. These error values are presented below in a table. The grey colored rows do not show considerable errors, whereas the remaining sequences exhibit large errors. From figures shown above, the following observations are made. Among the 43 sequences, 17 sequences do not show wide differences. Among the 43 sequences, 26 sequences show wide differences. Among the 87 sequences, the following 2 sequences show wide differences, S26 and S32. Now, among the 130 sequences, 85 sequences do not show any difference. Based on GR values, 130 virus sequences are classified in 4 groups. Number 1, S1 to S87 other than S26 and S32 show remote mutation possibility. Number 2, 26 sequences show very likely mutation possibility. Number 3, 17 sequences show very likely mutation possibility. And number 4, S26 and S32 show very likely mutation possibility. Apart from genome analysis, an effort has been made to devise solutions to dismember the structure of any given virus genome using sound frequencies. We call it as sonic attack. Or genetic modification could be carried out under the influence of acoustic, electromagnetic and scalar waves, meaning genetic code of DNA could be read and possibly rewritten. Nobel laureate Luc Montagnier has demonstrated that DNA can be generated by teleportation through quantum imprint, and also showed that DNA emits electromagnetic signals that teleport the DNA to other places, such as water molecules, a shocking revelation. The mathematical model for vibration behavior analysis of DNA and using a resonant frequency of DNA due to Mobin Marvi and Majid Gadhiri is given by two governing equations of DNA considering the effects of the cellular fluid and temperature change. In this slide, we see equations 1 and 2 as models to determine DNA natural frequencies. Some of the observations made are: number 1, as the DNA length increases, the natural frequency decreases. This need not be true in all cases because the natural frequencies depend on various factors like helical turns, viscosity, density and temperature of the fluid surrounding the RNA. Number 2, it is expected that the natural frequency associated with the coronavirus RNA could be of the order of kilohertz. What we try to make out here in this slide is that sonic frequencies could be artificially generated to dismember coronaviruses, a significant direction in devising methods to combat viruses. The 7 swaras, that is tones, of the saptak, that is set of 7 tones, are the fundamentals of heptatonic scales in Carnatic and Hindustani classical music, and they are Shadja, Rishabh, Gandharva, Madhyama, Panchama, Daivata and Nishada. Shadja is realized as the fundamental frequency Fs, which may be in any suitable scale. Then the remaining swaras, tones, are realized as the frequencies given by the following equations. Fr is equal to Fs plus 33 Hz. Then Fg is equal to Fs plus 69 Hz. Then Fn is equal to Fs plus 88 Hz. Then Fp is equal to Fs plus 131 Hz. Then Fd is equal to Fs plus 179 Hz. Then Fn is equal to Fs plus 233 Hz. And the surja of the next octave, we call it as Fss, which is equal to Fs plus 254 Hz. For example, if one chooses 50 Hz for Fs, then Fr is 83 Hz, Fg would be 119 Hz, Fm would be 138 Hz, Fp would be 181 Hz, Fd will be 229 Hz, Fn would be 283 Hz, and the shadja of the next octave Fss would be 304 Hz. For convenience, 4 frequencies such as Fs, Fm, Fp and Fss are chosen and allotted to 4 nucleotides such as adenine, thymine, guanine and cytosine respectively. The waveform of the audio signal is described mathematically as shown in this slide. In our case, A of t is a constant and the audio burst is on for time tau. The function of time is determined by the pulse repetition frequency and the duty cycle. The frequency spectrum of such a sequence of rectangular pulses is represented by a sinc function. This slide shows frequency spectrum of a sequence of rectangular amplitude modulated pulses. The efficacy of this technique of deactivating virus genome by sonic attack has to be checked real time by transmitting generated pulses to the actual target genome and by observing the changes in the shape of the genome. The control parameters are pulse width, pulse off time, amplitude of pulse and frequencies embedded inside the pulses. Apart from this, phase shift keying could also be incorporated as a part of intra-pulse modulation scheme to improve efficacy of the system. The stream of frequency bursts with Fs is equal to 50 Hz, then Fm is equal to 138 Hz, then Fp is equal to 181 Hz and Fss is equal to 304 Hz, corresponding to the nucleotides adenine, thymine, guanine and cytosine in the virus genome S1 is presented below. The complete list of bursts is not shown here due to lack of space. Now let me demonstrate sample sonic attacks on 9 virus genomes. I am just playing the frequency stream used to deactivate 9 virus genomes. I sincerely thank Professor Hu and Professor Sergey Petkov for the opportunity given to me to present a keynote on virus genome analytics. Thank you very much.